Um, so in typical fashion, each speaker will present and then we'll have questions at the end. I would ask if you to please, keep yourself on silent and type questions into the chat. We actually, because Mark has to leave, he will field his questions first, if that's okay. So you can think of that when you're writing your questions. And I think just with that, just to welcome everybody for this particular seminar and to remind everyone that we still have two more, one in April and May, on the UCC Cancer Seminar Series. So Mark will be presenting first today. So I will introduce him and he'll present, and then I'll do a short introduction for Dr. Patrick Ford as well. So first up is Professor Mark Tagney. He is a professor in education for enterprise and head of the IED Hub at UCC, a principal investigator in cancer research at UCC, and also a funded investigator at APC Microbiome Ireland. So he has a PhD in microbiology and an MBA from UCC. And his research interests for a long while have included associations between bacteria and cancer, and development of microbes as cancer therapeutics, in silico protein design, and synthetic biology. He's worked internationally at various institutes and has served on executive councils for the International Society for Cell and Gene Therapy of Cancer, scientific community for the European Society of Gene and Cell Therapy, and the IACR, the Irish Association for Cancer Research, and vice president for the Irish Society for Gene and Cell Therapy. So he's on numerous editorial boards and international journals. I've known Mark for many, many years. I'm sure we're in for a very nice talk about his latest work and with his group. So when you're ready, Mark, you can go ahead and present your screen, and we're happy that you're here. Thank you.
Thank you very much, Kelly, and thanks, Oisín, and it's a pleasure to be here. I'm just going to give very broad brushstrokes about the type of stuff that the guys in my lab do and have been doing, and I'll leave all the cool stuff to Patrick to talk to you about after. So overall, I guess, as Kelly mentioned, the experience that we have in my lab over the years is bacteria and cancer, and we approach that from two different angles. But overall, the ethos, I think, from which we approach development of our technology is this title here, the what, the so what, the now what, which I'll try my best to explain. In terms of disclosures, I have nothing to disclose. So overall, I suppose we're using this coined term oncobiome, which everybody knows in this day and age that if you've got a bad microbiome, it's bad for your health. And cancer is included there. So the questions that we try to ask when we're doing our what investigations are where or when are the relevant bacteria involved in cancer. So everybody knows that the main concentration and abundance of bacteria in the body is in our gut. So is that the only game in town when it comes to cancer? Do we just focus there and ignore everything else? Or could there be some bacteria in healthy tissue prior to any tumor arising? Do we need to investigate that? Or if a tumor is there first, is there some bacteria arriving at that tumor opportunistically after? Is that relevant, or can we do anything about it? So these are the type of what questions we ask. Then in terms of the now what, well, if there are bacteria there, what might they be doing? Are they involved in oncogenesis? Are they involved in tumor promotion? Are they fetching any treatment that goes on? And once we know the what and then so what, that's all very well, but what are we going to do about it? So that's why we take this now what approach. So I'd like to say that I focus more on the so what and now what, but you can't really do that without doing the what as well. So we take all angles, I guess, when we're looking at that. So broadly speaking, what might bacteria do? Well, I suppose the main focus is on their influence on the immune system. And over time, we're learning more and more about that. But I mean, it's well established ever before the microbiome story, even that different bacteria can have different influences on different immune cells, both type 1 and type 2, and in terms of immune modulation. In terms of the cancer setting, um, this is a complex diagram, but it's just showing that it's not a simple story um, in terms of what these bacteria are doing. So it's multifactorial. You can have systemic immunomodulation in non-cancerous states. So you have to look at what bacteria are in a healthy person that might be doing something. They could be anti-tumorigenic if you've got a good microbiome, for example. Um, that, a good microbiome can reduce inflammation. You could also have immunomodulation in the tumor microenvironment, and these could be tumor suppressive. On the opposite side of the scale, if you've got let's say, a bad microbiome, then it could be pro-tumorigenic in terms of the inflammatory responses going on, and indeed it could be tumor promoting as the tumor grows, depending what bacteria are where they might actually be helping the tumor to grow. A lot of labs around the world, much bigger labs than mine, have looked at the gut microbiome in cancer patients and the effects on treatment. This is just somebody else's review that kind of very nicely summarized some of the previous studies in terms of variations in what microbes were enriched during immunotherapy. And again, it's showing that it's far from a simple story. It's not like anybody has been able to find one particular bug, which if it's there, it's bad, or if another one is there, it's good. It's not that simple. So the blue bands here simply are saying that these are lots of different genera or species of bacteria that are enriched when immunotherapy, specifically immune checkpoint inhibitor therapy, is working, is the blue. But similarly, they've also found lots of other genera of bacteria um, which are enriched during ineffective ICI treatment as well. So in terms of our own work from the lab over the years, taking what other people know, we look at the microbiota cancer, and we look at the what, so what questions and the now what questions. In terms of the what and so what, we have kind of two broad approaches. We try and work with patient material as much as possible, and we try and make these microbial maps of the patients. So the type of material we look at would be related to the type of question we're asking. So in terms of the distal bacteria, so looking at, let's say, the main concentration of bacteria in the gut, what type of bacteria are there? So we try and do sequencing of the bacteria to establish the profile in different sets of patients. And different sets of patients will be different cancer types. I suppose my own lab is pretty cancer agnostic. And in terms of the treatments, so yes, it's the ICI treatments are, I suppose, still the mainstay, but we also have an interest in chemotherapy in terms of what bacteria might be doing there as well. So that's the distal bacteria to the tumor. So we have a number of studies going on with different excellent clinicians around the country in different types of patient treatments and different types of cancer indications. One of, I suppose, the mainstays of the work that we do is funded through an SFI and Breakthrough Cancer Research funded piece of work that I do as part of a national consortium, which is Precision Oncology Ireland. And the work package that I lead is the microbiome profiling of patients for their stratification. So, we want to see what influence the bacterial profiles have on the patient outcomes, and ultimately to see if we can use these profiles as potential biomarkers for various cancer treatments to personalize treatment regimens. In terms of the local bacteria is kind of the other stream of work in the lab. I guess we were the first 10 or more years ago to go and show that actually there are some bacteria of different patterns in patient tumors. And we initially were looking at breast cancer patients just because of the easier logistics in collecting them, but we've also been looking at other cancer types as well. So we go and sequence the type of bacteria, and these days we've been going beyond bacteria to other types of microbes as well. to see what's there and to look at differences between malignant tissue and non-malignant tissue. So in terms of that intratumoral microbiota, this is actually how my lab first got involved in, I suppose, microbiome work. We were looking at opportunistic infections of tumors. So for many years and decades, people have been looking for a specific species of bacterium in cancer patients' tumors to correlate that that bacterium is causing that cancer. So this is a summary, more than 10 years ago now, we put this review together of what was going on back then. And yes, does seem to be the case in many tumors, that there is a kind of a causative correlation between certain bacteria, pathogenic bacteria, and certain types of cancers. And this is growing as well. In terms of oncogenesis being I suppose the initial hypothesis, so people are looking at a certain type of microbiome promoting cancers of different types and going beyond just a single bacterial species to actually having a various profiles which are associated with oncogenesis. So they would be there in healthy tissue before the tumor arrived, and the hypothesis would be that they're causing it. And this does stand up with a lot of the evidence that other labs are showing. We don't get involved in that style of work typically, and we're more looking at the opportunistic tumor inhabitants, because work that we were doing before our microbiome work was administering mice with tumors, giving them a systemic administration, normally with tailbone injection, of different types of lab bacteria. And we weren't the first to discover this. People have known for many, many decades that actually when you do that, depending on the bacterium you're using, they can often be cleared from everywhere in the body except the tumor. There's various hypotheses for that, but mainly it's because they hide from the immune system in the tumor, and they can get into it because of the disorganized leaky vasculature in there. A lot of bacteria like the low oxygen potential within tumors, and there's lots of nutrients which spill out from, especially necrotic parts of tumors as well. So this is just a typical mouse experiment from the lab over the years. This is a bifidobacterium, a non-pathogenic probiotic bacterium. And if you look at the black bars, they're viable counts of bacteria from subcutaneous mouse tumors. And over time, that's the only place that you see increase in numbers. The gray and white bars are from healthy organs, and you don't see any increase in numbers there over time. So bacteria replicate specifically inside in tumors in these models. We've done that in lots of different ways, lots of different tumors. Here's an example of, again, a subcut tumor in a mouse where we tag the tumor cells with luciferase. So you can see that in kind of green-blue, and we tag the bacteria with Lux, which is a different type of luciferase, and you can see that in orange, and it's kind of concentrated in this case within the center of the tumor because it's an anaerobic bacterium. And you can see the main vasculature supply to the tumor in the red there. You can see the anaerobic bacteria like to stay away from that. So as I said, we've done lots of different types of tumors. Anything we can get our hands on in terms of tumor models, we try it. It works in everything basically. It's I suppose my current way of thinking of it is the bigger the tumor, the better. And I suppose because of that type of work, we got good at examining bacteria in tumors. So initially we were looking at mouse tumors, but we had a lot of technology in the lab for going examining their growth patterns and their profiles over time. We also had to look at various models, including sheep as well, for large models, and that also works. So this then led us onto the patient work. Originally we were saying, okay, might there be the odd patient where there's opportunistic infection? So we went looking at the breast cancer samples and just sequenced the bacteria there. So much to our surprise, yes, we were able to find bacteria in patient tumors, and lots of different types of bacteria in all the patients, much to our surprise. And then we looked at even non-malignant tissue in cancer patients, also had a different profile, but nonetheless a profile as well. And even when we looked at healthy patients, breast tissue, so from breast reduction operations, even they had a pattern of bacteria as well, but very different to those from cancer brain patients. So there was something going on. So we kept on that stream of work in the lab and other people. around the world, other labs started finding it after us then as well. So that's now a kind of a relatively small field, but a field nonetheless in terms of the intratumoral microbiome in lots of different tumor types. We did realize, though, I suppose, because we were early in the game, that the microbiome technology moved on, and especially when we're looking at low biomass samples and tumors, that you have to be incredibly strict in terms of what's real and what's not. So we put a lot of effort over the years then in terms of improving the methodology from various angles, so in terms of the biological considerations and the bioinformatic aspects. So we needed to remove whatever isn't real to see if there's still something there. And there is. So we made a lot of different models and analysis strategies, both biologically and bioinformatically, and published them in different journals. So we're still confident, even though I didn't originally believe it. I suppose because we've gone so stringent now, I'm starting to believe that there's something going on, all right. So that led us to ask then kind of, if they are there, what are they doing? Is it relevant at all? So we started asking the so what questions. So we started looking at, can they potentially influence cancer treatment? Could we exploit them as biomarkers, or indeed exploit them in therapy? So with cancer treatment, we had a look at, if you think about a bacterium, any bacterium as a bundle of enzymes, could these enzymes be affecting any small molecules? So we did some screens of typically use cancer chemotherapeutics, because a lot of them are pro-drugs which are affected by enzymes anyway. And we did an in vitro screen. I found that some of them went up, a lot of them nothing at all happened, and some of them went down. So then we did some HPLC and mass spec to see if I could identify what was going on. So as you can see here, you can see the gemcitabine was being reduced in efficacy by the presence of some of the bacteria we were finding in patients. Some of the other ones were being increased, but we could see, especially by the mass spec, that it was biotransformation, it was enzymatic activity from the bacteria. And we could pinpoint it to what type of activity, like acetylation or hydrolysis or reduction. We replicated that in mouse models as well. So I suppose if you just look at the bottom survival curve there, you can see that if you have a mouse and you fill up its tumor with bacteria, you dramatically reduce the effect of the gemcitabine. So the green line is gemcitabine alone. So you do have prolonged mouse survival by treatment with gemcitabine. If you have bacteria and gemcitabine, the blue line, you reduce that way down. So the presence of the bacteria is switching off the gemcitabine drug. And we did that for some other drugs as well. So we published that originally just in scientific reports, and then other labs went and published it in much bigger journals than us, with bigger address universities than us. So then we moved into the now what. So can we do anything with that? Well, yes, in terms of bacteria, there's different strategies you could propose in terms of bacterial therapies, such as bacterial drug vehicles. You could do microbiome modulation with diet, probiotics, or FMT, or exploit these bacteria's microbial biomarkers for precision oncology. We can do some of these in terms of the biomarkers. Again, in terms of the patient work, we went and sequenced again fresh breast samples, and we trained using machine learning on certain data sets to see if we could come up with an algorithm whereby could this algorithm go and identify what's malignant tissue versus what's non-malignant tissue based on the microbial profile. And it worked. So we could see that actually, not that this will ever replace anything as a diagnostic or anything, but just as a kind of proof of concept, that the bacterial signature alone can tell what type of tissue it's in. And in terms of exploiting in therapy, well, our approach is, I guess, back to our original approach, which we kind of termed the bacterial biofactory. So using bacteria as drug delivery vehicles because they're selective for tumor tissue. So we, I suppose, have pretty good competence in the lab in terms of engineering bacteria, and we engineer smart bugs so that they can sense their surroundings, compute a response, and then go and respond by producing something appropriate. So we've done that with a lot of different types of drugs, but in our case, it's proteins. So what type of proteins are produced by the bacteria in terms of immune therapy or chemotherapy activation, re-educating macrophages. Overall, we try and think of it as conditioning a tumor. So if you can fill up a tumor with bacteria, and then you can get your bacterium to do something, you can condition the tumor in a certain way. So overall, to sum up, I think we're better equipped now than we were some years ago in terms of the field. So we know more what and so what because of the microbiome studies. We've got improved methodology. In terms of, I think we can rely more on what we're finding in tumors now in terms of bacteria, and we've got enabling technology by way of synthetic biology and protein design for turning these bugs into drugs. So thank you very much to all my funders and to everybody for listening today. Thank you.
Thanks, Mark. That was perfect. I like your statement of now I think I might believe it after all these papers and literature. So I would just like to remind the audience to, if you have questions for Mark, to please type them into the chat because we're going to move on to the next speaker. And I will introduce him while he gets his slides going and everything. So this is Dr. Patrick Ford, who's joining us today. He's currently a professor and co-director in the Division of Upper Aerodigestive Malignancies in the Department of Oncology at Johns Hopkins University. He directs a multidisciplinary thoracic oncology clinical research program there. He's led the development of clinical translational research program focused on the immuno-oncology of the upper aerodigestive malignancies. Dr. Ford's research examines the role of immunotherapy from mesothelioma. I'm sorry, I'm just tired. The meds, I just lectured to med students and they were killing me. And lung cancer. And his work has led to the development of several ongoing phase 3 trials. In 2022, his work over several years was published in the New England Journal of Medicine and led to the FDA and EMA approval of neoadjuvant chemotherapy for the treatment of surgically operable lung cancer. Dr. Ford, we're very happy to have you join us today from Johns Hopkins. I assume you're there in Maryland.
Sure.
And you're very welcome. And when you're ready, just share your slides and we look forward to your presentation. Thank you.
Perfect. Thanks very much, Kelly. It's great to be here. And I enjoyed Mark's presentation a lot. I'm just going to pull up the slides. Just one second. Perfect. So I'm going to give a little bit of background about the neoadjuvant immunotherapy and then talk about some of the potential biomarkers or predictors of benefit and lack of benefit. These are my disclosures. So there's a lot of a discussion about the best way to reduce cancer mortality in general. And for many years, I think it was recognized that a reduction in, so prevention is better than cure, and that's still true, I think. So things like reducing smoking incidence, early detection of cancer as well. However, we're starting to see some of these systemic therapies actually have an effect on population mortality. And this was a nice paper from Dr. Howlatter and colleagues from the National Cancer Institute a couple of years ago now, where she looked at the reduction in lung cancer mortality over a 5-year period and compared that to the reduction in incidence over the same period. And historically, lung cancer incidence came very close, unfortunately, to lung cancer mortality because most patients who developed lung cancer actually passed away from it. But that's been improving, and we're starting to see now an actual reduction in all-cause mortality in lung cancer, likely due to new systemic therapies. There was a period over 5 years from 2015 to 2020 where there were more than 30 new lung cancer approvals. And since 2020, there's been another 17. So it's a dramatically changing landscape. And hopefully some of these figures you see on the left will improve as a result. When we talk about early stage lung cancer, this is about 30 to 40% of patients. And these are patients who don't have metastasis of the cancer outside of the chest and are generally considered either stage 2 or stage 3 disease. Despite this being cancer which hasn't spread distantly, the prognosis historically was still not good, ranging from a 5-year survival of about 36% for stage 3 disease to 60% for stage 2. And kind of a rule of thumb here on the right is when you're talking about these cancers in lung cancer, you're talking about a tumor that's either 4 centimeters or greater in diameter in the lung, or it's involving a lymph node or both. This is about 500,000 people worldwide. Here in the US, it's about 40 to 50,000 people each year have these stages of cancer. So we know surgery can cure roughly 50% of people, which is lower than most other common cancers, but can systemic therapy make a difference? There are 3 different ways really of giving systemic therapy around the time of surgery. On the right of your screen here, you'll see adjuvant, which is giving it post-operatively. So the surgeon removes the cancer and then the patient gets a systemic drug treatment post-operatively. On the left, you'll see neoadjuvant, which is giving the therapy prior to surgery. And this might have potential benefits in that patients in general haven't been through the physical trauma of surgery, so more patients will commence the treatment, more patients will complete it. And in the era of immunotherapy, we're seeing that having the draining lymph nodes where the tumor drains to essentially in the middle of the chest, still in situ, where they haven't been removed by the surgeon, potentially has benefits. And this has been shown in a randomized trial in melanoma, that those patients who get neoadjuvant immunotherapy compared to adjuvant tend to do better. From a scientific standpoint, there's also potential benefits in that when we're trying to do some of the translational science and so similar analysis to those Mark described in the tumor, for metastatic disease at the bottom left here, you're dealing with a very small core biopsy, generally 1 millimeter or less in diameter, and anything from 5 millimeters to 10 millimeters in length. Whereas here on the right side, you have a longer section after neoadjuvant therapy, and you can imagine the quantity of tissue available for functional and for in vitro analysis are much greater. We did a small study now about 5 years ago looking at just 2 doses of immunotherapy, so an immune checkpoint inhibitor called nivolumab. Nivolumab blocks PD-1, which is a co-inhibitory immune checkpoint, so essentially downregulates the immune response to antigen. And here, when you gave nivolumab to patients prior to surgery, we were surprised even just 2 doses over 4 weeks prior to surgery led to fairly significant tumor regressions, about half of the patients having little or no residual cancer at the time of resection. You'll see some of the cases here on the right. And the right upper panel there shows a large tumor in the lung. You see the big white area in the middle with the arrow pointing to it. After 4 weeks later, at the time of surgery, the tumor had involuted a lot and there was no residual viable cancer. On the bottom right here, you'll see a different pattern of response. So in panel B there, you'll see the arrow pointing to a large right upper lobe tumor. The tumor appeared to have increased in size 4 weeks later. However, when that tumor was removed, it again didn't have any residual viable cancer, but rather had a lot of infiltrating immune cells. So you can see different patterns of response with these drugs. We're also able to show that at least in this small study, those patients who had higher non-synonymous numbers of tumor mutations present were more likely to have a pathologic response. On the left of your screen here, you'll see a multiplex immunofluorescence image showing in the orange area is an area of tumor. You see very few CD8 positive green cells infiltrating it prior to nivolumab. Those that are present are co-expressing PD-1 in red. However, after treatment, so 4 weeks later at the time of resection, you see the tumor has been infiltrated by a lot of CD8 positive T cells, leading to tumor regression and a major pathologic response. There's a lot of potential translational science you can perform in conjunction with neoadjuvant therapy because you have that very early response indicator of pathologic regression. And here we looked at clearance of circulating tumor DNA prior to surgery. So over that 4 to 6 week period, here you'll see in red a specific mutation here, a P53 mutation, which was detected in plasma, so in blood prior to treatment. It went away with treatment. Here you'll see in green is the radiology. So there was a slight decrease in the size of the tumor on scans. However, when the tumor was removed, there was no residual viable cancer. So In this case, checking ctDNA in peripheral blood tracked more closely with the pathologic response. And on the other hand, you can see here where the KRAS and an ALK alteration, when they increase, that also seems to track well with the lack of tumor response here in red, very little pathologic response in this case. We've now followed these patients for over 5 years, and those patients who had a major pathologic response, which was defined as 10% or less residual viable cancer, have done very well in general. No patient has had active cancer at the time of report, whereas unfortunately those patients whose tumors didn't respond so well, multiple of those patients had a recurrence of their cancer, and some died from their cancer. So this is a potential early surrogate. The idea of the neoadjuvant model, particularly in the context of immunotherapy, lends itself very well to translational science. These are just some of the work from that initial trial back in 2018. And it involves a team of scientists, research assistants, close integration with the clinic, with surgeons, medical oncologists, and radiation oncologists, in order to bring all this to meld the clinic and the lab together. Some of the work our group has continued to do has been particularly focused on functional immune analysis and single-cell RNA-seq, where we can get fresh tumor from the operating room, from the operating theater, and digest that tumor in the lab, and then abstract the live immune cells from those tumors, and then potentially perform single-cell RNA-seq if there's a sufficient number present. One of these analyses has actually led to a further clinical trial, and this was a study published by my colleagues, Dr. Kowsey and Dr. Smith, in Nature 2 years ago now. And this was looking at mana-specific T cells, so T cells which are recognizing mutations in the tumor and are attacking the tumor as a consequence. Can you profile those isolated T cells using single-cell RNA-seq? Essentially, those tumors which were responding well to nivolumab, the mana-specific TIL from those tumors expressed T cell receptors with markedly higher ligand-dependent signaling, and also in particular higher levels of IL-7R. And we've been working with a company called Compugen to develop a clinical trial with an agent which may raise levels of IL-7R. Others have continued on these studies. There's been 2 major phase 2 trials, one conducted by Dr. Provencio and colleagues in Spain called Nadim, and this looked at giving 3 cycles of chemotherapy along with nivolumab prior to surgery. Showed pretty remarkable results. I would direct your attention here to the pathologic complete responses. More than half of the patients, 63%, had no residual viable cancer at the time of tumor resection. And here on the right, here in the US, Dr. Kathy Hsu conducted a similar study with an anti-PD-L1 antibody, atezolizumab, which showed 1 in 3 patients had no residual viable cancer. And when you look at the historic data with chemo alone, you're looking at just 2 to 6% of patients having no residual cancer after chemo. So the immunotherapy here is having a very dramatic effect. We conducted a study called Checkmate 816, which was a randomized phase 3 trial with the intention of teasing out this question of whether the addition of anti-PD-1 immunotherapy to chemo prior to surgery could potentially improve survival long-term. This randomized patients with newly stage 1 to 3 cancer to a control arm of 3 cycles of standard chemotherapy or chemo plus for 3 cycles prior to surgery. The surgery was planned within 6 weeks of the last cycle. And so in contrast to some of the other trials in this space, there was no further mandated treatment post-operatively. So patients could get further chemo, but they didn't receive further immunotherapy. So this is a short course of treatment. Primary endpoints were pathologic complete response and event-free survival, both by blinded independent review. Event-free survival means essentially it's a composite endpoint looking at, do you get to surgery? Do you have a recurrence of cancer? Or do you die from your cancer? And can we improve that number for patients with the addition of nivolumab? These were the characteristics. So you'll see about half the patients were from Asia, about half the patients had squamous disease, And close to 2/3 of the patients had stage 3 disease, so relatively locally advanced. You'll see here that the large majority, 94% and 85% in each arm, completed the planned neoadjuvant treatment. This, I think, is in contrast to when you give treatment postoperatively, patients are less likely to complete the actual treatment. 83% of patients in the nivolumab plus chemotherapy arm had definitive surgery, and actually higher numbers in the control arm got some adjuvant treatment. Somewhat unusually, when you add a drug to a standard therapy, we didn't actually see any significant increase in toxicity, perhaps because of the short course of treatment. The first endpoint which matured was pathologic complete response, and this showed a significant improvement with the addition of nivolumab here on the left. Among all patients included in the trial, it went from 2.2% with chemo alone to 24% with nivo plus chemo. And now we followed these patients for over 3 years. And the other key endpoint of event-free survival is also significantly improved from 43% in the control arm at 3 years to 57% in the investigational arm. And this led to the approval of nivolumab plus chemotherapy initially in the US and now in close to 30 countries worldwide. You'll see here are there groups who benefit more? Well, it does appear as if those patients with higher stage tumors derive more benefit. Also, those patients whose tumors are PD-L1 positive. However, in general, most of these bars are to the left of unity. So if a patient does experience a recurrence, what is the effect of nivolumab? So here we see that the big effect here of nivolumab is actually in reducing distant recurrence, particularly in the brain. So you'll see the incidence of brain metastasis in the control arm with no immunotherapy is 13%, and that's reduced to 4% in the investigational arm. In contrast, there isn't as much of an impact in the chest itself. So this is primarily an effect on distant recurrence, which does account for the majority of deaths from lung cancer. An interesting finding as well was that, so you might expect when you add a drug to a standard treatment, could the surgery become more complicated? Apologies. But in this case, that didn't happen. Patients were less likely to require a pneumonectomy, which is removal of the whole lung, if they got immunotherapy. And they were more likely to be able to have a lobectomy, which is lung-sparing surgery. We looked at various translation science analysis here. This is a 4-gene inflammatory signature from the baseline tumor biopsy prior to any treatment. And this appears to predict benefit in those patients who got immunotherapy. So it's predictive when you get an immune-based treatment, not predictive for chemotherapy, is essentially what this slide is telling us. And it's also predictive in terms of event-free survival. So you'll see here on the left of the screen, in those patients who got nivolumab plus chemotherapy and had the baseline inflammatory score, they were more likely to have a prolonged event-free survival, having a positive 4-gene score versus not, whereas it didn't appear to make a difference for chemotherapy. I mentioned earlier a few anecdotal cases of looking at circulating tumor DNA. Here is a more in-depth analysis, and this was looking at using tumor-informed circulating tumor DNA. So this involves whole exome sequencing of the baseline tumor biopsy and creating a panel based on that whole exome sequencing specific to the patient, and then tracking those alterations in plasma over time. Those patients who, during the neoadjuvant phase, so during the 3 cycles of preoperative therapy, if they cleared their ctDNA versus didn't clear, there was a trend, not significant because of the numbers, but a trend towards improved event-free survival. We now have 3-year follow-up on overall survival. It's not fully mature yet, despite the p-value, but the hazard ratio is trending significantly in favor of nivolumab plus chemotherapy, with about a 14% difference between the 2 arms at 3 years. Sorry, keep getting want me to go back to work. And also, one potential very simple biomarker would be looking at pathologic complete response. So when you remove the tumor, is there viable cancer remaining or not after neoadjuvant therapy? Those patients who had a pathologic complete response appear to do very well. And so as a reminder, in the nivolumab plus chemotherapy arm, this is about 24%. And essentially, they have a high likelihood of having event-free survival at 3 years, about 90% or so, and more than 95% of patients who have a pathologic complete response are alive at 3 years. So you could imagine scenarios where for those patients who don't have a pathologic complete response, you might change their therapy post-operatively or intensify their treatment in terms of other clinical trials. So as I mentioned, this has been approved by the FDA now just over 2 years ago. So as of last August, it was approved in 25 jurisdictions. I think they're up over 30 now. And cost effectiveness, I think this is one potential benefit of this regimen over some of the others, in that it's 3 doses of treatment. So the National Institute for Clinical Excellence and NICE, which is the cost effectiveness body in the UK, assessed and neoadjuvant chemo-nivolumab as more cost effective than either neoadjuvant or adjuvant chemo alone, chemoradiation followed by surgery, or surgery alone for clinical stage 2 or 3A non-small cell lung cancer. We also, in the same trial, I'm not going to dive into it in too much detail, but we had a third arm looking at combination immunotherapy. So can you omit the chemo? And essentially what that arm showed was that overall, Nivo plus Ipi, 2 immune drugs, was better than chemo alone. But you did have quite a few patients had early progression on treatment. However, it would be nice to have a regimen where you spare chemotherapy, and perhaps using the 4-gene inflammatory score could be helpful here, in that those patients who had a high score, so on this inflammatory signature, had a prolonged event-free survival compared to those who did not on the left of your screen. There are several other trials ongoing, 3 of which have now reported. And these other trials are all looking at perioperative treatment. So Checkmate 816, the one I described, was just neoadjuvant, just 3 cycles of treatment preoperatively. All of the other trials have preoperative treatment and postoperative treatment. And 3 of those trials now, Keynote 671, Aegean, and Checkmate 77T, are all positive for the event-free survival endpoint. And one of those has been approved in the US and recently approved in Europe by the European Medicines Agency, which I'll mention briefly. This was Keynote 671. So this again is kind of pushes the criteria for surgery here to 3B. So more locally advanced tumors, where the tumors involving multiple lymph nodes in the chest, randomized to 4 cycles here of standard chemo plus placebo, or chemo plus pembrolizumab, which is an anti-PD-1 antibody. The big difference here is that in the postoperative setting, you get about 9 to 10 months of pembrolizumab postoperatively. These were the characteristics of the patients, and you'll see here, this is starting to push the envelope in terms of actually what patients are surgically resectable, with many patients here with 3B disease enrolled, almost half of the patients having mediastinal lymph node involvement. And this study also showed a significantly improved event-free survival, favoring the pembrolizumab arm, hazard ratio 0.59. And again, you see this separation by pathologic complete response, where those patients who have a PCR do significantly better than those who do not. One argument perhaps for the perioperative approach is that in those patients who don't have a PCR, you see some separation here. So this is potentially, in the absence of a clinical trial, is potentially one argument for giving the postoperative pembrolizumab. And this trial recently did mature for overall survival and significantly improves overall survival over placebo alone, showing a hazard ratio of 0.72. And this is actually the first trial in early stage lung cancer to show an overall survival benefit for about close to 20 years at this point. We have moved on to several other trials. So we conducted a trial called NeoCOAST, which looked at administering more of a window of opportunity trial, where you're giving a single immune drug, durvalumab, plus novel agents, so no chemotherapy, over 1 month prior to surgery. So mimicking the very first trial we did, but adding multiple novel agents in a platform design. And here, and this also allows you to look kind of in depth at the translational science. In contrast to our initial trial, tumor mutational burden didn't appear to correlate significantly with a pathologic response in this trial. However, several patients who had alterations which are classically described as resistant immunotherapy did not have a major pathologic response, and this is something we're going to continue to watch. There was also a suggestion that those patients who were highly inflamed, so had a positive gene signature for intratumoral immune activation, were much more likely to have a pathologic response. So mimicking here some of the CheckMate 816 data. This study was recently published, actually a nod to the microbiome here, and those patients who had greater stool taxonomic richness were more likely to have a major pathologic response across all arms enrolled in the trial. We've moved this on now. That trial completed accrual about a year and a half ago. We've moved on to another study called NeoCOAST-2, which here is trying to add on to the current standard of care, which is chemo plus immunotherapy. And you can see here multiple arms are enrolling. This study is active in Ireland. Dr. Juriskin is the PI. And essentially here as well as getting the novel agents, so the oleclumab, monalizumab, etc. Preoperatively, the patients will also get them postoperatively. So finally, where do we stand with these treatments? So it appears that the addition of anti-PD-1 immunotherapy to neoadjuvant chemotherapy so is becoming the preferred option. So for lung cancer, I think it's been included in most of the guidelines now as the preferred, that patients should be assessed for neoadjuvant therapy preferentially. We don't have head-to-head data, however, and it does require quite a substantial change to our clinical workflow, and that the patients frequently will see a surgeon or a respiratory physician first, so they need to be referred to medical oncologists for this to be implemented. All patients have to have resectable cancer up front, so we should, in our multidisciplinary meetings, be deciding up front whether the tumor is resectable or not. And we have some kind of indirect data to suggest that perioperative pre and post treatment may have some benefits, but it hasn't been directly established as yet. So I think that's everything, and I'm happy to chat or take any questions if I can stop sharing.
Yes. Thanks so much. What an amazing message, I think hopeful. So I put into the chat for questions, and Professor Mark Tagney must leave a little bit before the hour, so if you have anything for him, let me know. But there's a question in for Patrick first from Rasheen. Can you comment on whether baseline biomarkers, archival tumor biopsies, or biomarkers from residual surgical specimens may be most helpful in this neoadjuvant immunotherapy setting? Both are challenges with either.
Yeah, so I think ironically the most so in contrast in advanced lung cancer, we have several biomarkers we look at. PD-L1 is probably the most commonly used one, and is a relatively simple immunohistochemical marker. We also look at advanced lung cancer at multiple mutations for targeted therapies. But PD-L1 in advanced lung cancer doesn't appear to affect the efficacy of chemoimmunotherapy when you're giving immunotherapy with chemo. But in early stage lung cancer, it does appear to be predictive in all of the trials so far. Those patients who have higher PD-L1 tumors do seem to derive more benefit. So that's probably the key simple biomarker we're looking at. There's also, I think, the dynamic biomarkers, like I mentioned, ctDNA, and those early changes you could imagine could be very helpful. If you're a surgeon, for example, and you see the patient back 9 weeks after starting the therapy, and the tumor sometimes doesn't shrink, it stays the same, or it even gets a little bit bigger, but you had a ctDNA which is completely cleared, I think that would give you some flexibility when you're doing the operation. Are you going to try and push to do a full pneumonectomy, or will you be happy with the lobectomy? And I think some of the surgeons are integrating those into kind of the thought algorithms to start with. And then there's multiple other things. I mentioned microbiome is being looked at in stool and also in the resected tumors. And I think some of these 4-gene inflammatory scores at baseline, they're still not, despite being looked at now in both advanced disease and early stage disease, they haven't been validated in kind of prospectively. And I think that's the big challenge in this. We need prospective validation for many of the biomarkers, which we don't yet have. So PD-L1 is about the only one which is routinely used at the moment. And also most of the trials excluded patients with mutations. So we do at a minimum need to test for EGFR and ALK in lung cancer too, because those patients would not go on this treatment.
Okay, perfect. I don't see any questions for Mark, but I might ask one if he's still around. So your synthetic biology, I mean, I'm always very curious about this. So if you can engineer your favorite microbe to make something, like how far it, like at the minute, maybe it seems like a little sci-fi, but like how far are we away from kind of really widespread interventions like this to help cancer patients, do you think?
Yeah, it's a great question, and I wish I could say we're not very far, but you know, I guess the design of these bugs, from a technical perspective, you could say it's wide open and you can do pretty much everything. But some might argue, but the design has to be informed by, you know, patient need, but probably more importantly, unfortunately, regulator requirements. So I think it's down to the progression will be driven more by, let's say, the progression of the regulators' understanding of live biotherapeutics and ATMPs. And that's increasing a lot in recent years. So, you know, a number of years ago, it would have been a flat no, you can't do a clinical trial on something heavily engineered derived from a pathogen. But as time goes on and other similar live biotherapeutics are approved, then the knowledge of the regulator becomes better and more things are coming along. So like I've always followed the field of oncolytic viruses because they're very similar. You know, they're genetically modified, originally pathogenic microbes. And once the first one of them was approved, I think that's been paving the way for bacteria as well, and there's other, I suppose, precedents as well. So from a technical point of view, I won't say the world's our oyster, but there's a balance between, you know, the delivery bug, the producing bug, and what it's delivering. So we have put a lot of time recently, I didn't speak about it in my talk, but into in silico protein design, because it was overly simplistic, just having an old-fashioned approach of putting a human gene into a bug and hope for the best with the protein. So I think the synthetic biology that we focus on more is these designer proteins inside the designer bugs. And I think that brings another regulatory challenge because it's no longer native protein. So it's something brand new as far as the regulator is concerned. But I think they go hand in hand. So from a technical perspective, everything's going very quickly. But from a regulator's perspective, it just has to wait a little bit, but hopefully soon.
Okay, thanks so much. There's one more question for Patrick. This is in the chat. Does PET or performance status help differentiate true versus pseudo progression in patients with enlarging tumors following the neoadjuvant treatment?
Yeah, the PET is interesting in that you can actually see quite a few patients, it's been pretty well described, have what's called nodal immune flare, where you get increased uptake on PET scan, so in the mediastinum, so where the lymph nodes are, and that several groups have resected those lymph nodes and found no viable cancer. So in general, I think standard FDG PET is unlikely to be helpful in terms of differentiating true versus pseudoprogression. And those patients who have a better performance status overall are more likely to benefit from immunotherapy or from systemic therapy in general. And all of these trials so only kind of enroll those patients who are not symptomatic or minimally symptomatic. So that's kind of an important point to keep in mind when we're discussing patients in the tumor boards as to whether they're candidates for neoadjuvant therapy. But in general, I think there's a lot of work to try and predict which patients will benefit most and minimize toxicity as well.
Okay, thanks for that. I think we might leave it at that because I'm probably going to be chased out of the room that I
Mhm.
chased other people out of to get in here. Uh, so I just want to thank again
Thanks,
Professor Mark Tagney and Dr. Patrick Ford. Thank you so much for joining us for really super informative talks today. We're back again with the Cancer Research Seminar in April. It's on the 15th, Monday the 15th. Thanks to everybody for attending as well and for the questions. And we'll see you again in April. All right, take care. Bye.